Hello my dear students, welcome back to Biology at Priyajit. Today we will discuss a very important topic of plant biology, that is photophysiology of plants. In this class we will cover basic photoprocesses, light trapping mechanisms, photophosphorylation, carbon dioxide fixation pathways, C3, C4 and C2 pathways, photomorphogenesis, plant photoreceptors. So let us begin. What is photophysiology? Photophysiology is basically refers to the study of how light affects plant physiological processes or physiological processes in plants. It mainly includes light absorption by pigments, energy conversion during photosynthesis, photophosphorylation, carbon fixation pathways, photorespiration, light regulated plant development or photomorphogenesis. Light acts in plants as energy source for the purpose of photosynthesis, information signal. It stimulates growth and development. Plants can detect light intensity, light quality or wavelength of light, light duration, and light direction. Basic photoprocesses. Photosynthesis basically begins when light interacts with pigment molecules inside chloroplast. The major photosynthetic pigments are chlorophyll A, chlorophyll B and carotenoids. When chlorophyll absorbs light energy, electrons becomes excited and move to a higher energy level. This excited energy can be released as fluorescence, heat, or can be used as photochemical reactions that drive photosynthesis. Energy absorbed by accessory pigments is transferred to reaction center chlorophyll through resonance energy transfer. So this is the image showing the absorption of light, wavelength of light by carotenoid, chlorophyll A and chlorophyll B. Chlorophyll absorbs mainly blue light in the range of 430 to 450 nanometer and red light in the range of 640 to 680 nanometer. So green light is reflected, therefore plants appear green. Excitation of chlorophyll. We know that when chlorophyll absorbs a photon, an electron becomes excited. Electron moves from ground state to excited state. Then excited state is unstable and energy must be released. Fate of the excited energy. The excited chlorophyll molecules may lose energy through fluorescence, that is light energy is released as light, heat dissipation, energy released as heat, and photochemical reaction, energy is used to drive photosynthesis. Only photochemical reactions lead to photosynthesis. Resonance energy transfer. Light energy is absorbed by one pigment and can be transferred to another pigment without electron transfer. This process is called resonance energy transfer. Energy moves through antenna pigment until it reaches the reaction center. Light trapping mechanism. Light harvesting occurs in photosystem located in the thylakoid membrane. The light energy is captured by light harvesting complexes present in the thylakoid membrane. Two major photosystems operate during photosynthesis, such as photosystem 2 with reaction center P680 and photosystem 1 with reaction center P700. Photosystem 2 splits water molecules, releasing oxygen, electrons and protons. Electrons then pass through an electron transport chain to photosystem 1. Light harvesting complex or LHC is composed of chlorophyll A, chlorophyll B, carotenoids and proteins. Its function is to capture protons and then transfer excitation energy to reaction center chlorophyll. Photosystems, there are basically two photosystems operate. Photosystem 1 produces NADPH and transfer electrons to NADP plus, and photosystem 2 or PS2 splits water, produces electrons and release energy. Photosystem 1 reaction center is P700 and absorption peak is 700 nanometer. Photosystem 2's reaction center is P680 and its absorption peak is at 680 nanometer. So both of them are located in thylakoid membranes of chloroplast. Jet scheme of electron transport chain. The jet scheme explains the flow of electrons in photosynthesis. Electron flow: water to PS2, then plastoquinone, then cytochrome complex, then plastocyanin, PS1, then ferredoxin, and then NADP plus. The products formed are ATP, NADPH, and oxygen. Photolysis of water occurs in PS2 and the reaction is 2H2O is converted into 4H plus 4 electron plus oxygen. Importance: this jet scheme electron transport chain is the sources of electrons It releases oxygen and creates proton gradient for ATP synthesis. So what is photophosphorylation? Photophosphorylation is basically the synthesis of ATP using light energy. During this electron transport process, a proton gradient is formed across the thylakoid membrane, and this proton gradient drives the energy, drives the enzyme ATP synthase, which synthesizes ATP. This process is called photophosphorylation. Two types occur: non-cyclic photophosphorylation producing ATP, NADPH, and oxygen, and cyclic photophosphorylation producing only ATP. Now, non-cyclic photophosphorylation occurs through photosystems. Both photosystems, light excites pH2, then water splits, electron moves through electron transport chain, then proton gradient is formed, ATP is synthesized by the enzyme ATP synthase, and pH1 produces NADPH. Here the products are ATP, NADPH, and oxygen. Cyclic photophosphorylation occurs only in pH1. Here the electron path is pH1. From pH1 electron is transferred to pyridoxine, then cytochrome complex, then plastocyanin, and then again pH1. So here ATP only is the product. No oxygen or NADPH is produced. It maintains ATP or NADPH balance. Carbon dioxide fixation or Kelvin cycle. Carbon dioxide fixation occurs in the stroma of chloroplast. The cycle consists of three phases. The ATP and NADP generated during light reactions are used in the Kelvin cycle for carbon fixation. In this cycle, carbon dioxide combines with RuBP through the enzyme RuBisCO. The first stable product formed is 3-phosphoglycerate, which is why the pathway is called as C3 pathway. Phase 1 carboxylation. Carbon dioxide reacts with ribulose 1,5-bisphosphate or RuBP. The enzyme is RuBisCO. Product here is the two molecules of 3-phosphoglycerate or 3-PGA. Phase 2 is reduction. 3-PGA is converted to glyceraldehyde 3-phosphate or G3P using ATP and NADPH. Phase 3 is the regeneration step. Here RuBP is regenerated so the cycle can continue. Energy requirement to produce one glucose molecule, 18 ATP and 12 NADPH is produced. C3 pathway. Now let us understand the C3 pathway of photosynthesis. The C3 pathway, also known as the Kelvin cycle, is the most common pathway of carbon fixation in plants. In this pathway, the first stable product formed is 3-phosphoglycerate, which contains 3 carbon atoms, and therefore it is called the C3 pathway. This pathway occurs in many important plants such as wheat, rice, and barley, as well as in most temperate plants. The key enzyme involved in this process is RuBisCO, which helps fix carbon dioxide during the Kelvin cycle. However, RuBisCO has a dual activity. It can act as carboxylase, fixing carbon dioxide for photosynthesis, or as oxygen is reacting with oxygen instead of carbon dioxide. When RuBisCO functions as oxygenase, a process called photorespiration occurs, which reduces the efficiency of photosynthesis. Therefore, although the C3 pathway is the most widespread carbon fixation pathway in plants, it has the initiation of photorespiration under certain environmental conditions. So during C3 pathway, the first stable product is phosphoglycerate, which is a 3-carbon compound, and this pathway is found in wheat, rice, barley, and most temperate plants. The problem here, RuBisCO has dual activity, carboxylase and oxygenase. When oxygenase activity occurs, photorespiration is continued. C4 pathway, are also known as Hertzsprung pathway, according to the names of the inventors of this pathway, and it is an adaptation to high temperature and intense light conditions. In some plants such as maize and sugarcane, carbon dioxide fixation occurs through the C4 pathway. Here CO2 or carbon dioxide is fixed first by PEP carboxylase or phosphoenol pyruvate carboxylase to form oxaloacetate. This mechanism helps prevent photorespiration and increases photosynthetic efficiency in hot climates. The key features of this pathway here: first, carbon dioxide acceptor. Carbon dioxide acceptor is PEP or phosphoenol pyruvate, and first stable product is oxaloacetate, which is a 4-carbon compound. The steps are: carbon dioxide fixed in mesophyll cells by PEP carboxylase, then oxaloacetate is converted to malate, then malate is transported to bundle seed cells, and carbon dioxide is released for Kelvin cycle. The significance are following: it prevents photorespiration, higher photosynthetic efficiency, adaptation to tropical climates, etc. C2 pathway, which is also known as photorespiration. Sometimes RuBisCO reacts with oxygen instead of carbon dioxide. This leads to photorespiration, also called the C2 pathway. Photorespiration reduces photosynthetic efficiency but helps protect plants from excess light energy. The cytopathy occurs when RuBisCO acts as oxygenase reaction RuBP plus oxygen to produce 3 PGA, 3 phosphoglyceric acid and phosphoglycolate. Organelles involved here, photorespiration occurs across 3 organelles. Number one is chloroplast, then peroxisome, then mitochondria. Effects: it consumes ATP, it releases carbon dioxide and reduces photosynthetic efficiency. Possible functions: it protection from excess light, detoxification of phosphoglycolate, and maintenance of metabolic balance. Photomorphogenesis. Apart from photosynthesis, the light also regulates plant growth and development. This process is called photomorphogenesis. Examples include seed germination, leaf development, and flowering. Photomorphogenesis is basically the light control plant development independent of photosynthesis. Plant photoreceptors. Plants detect light through specialized photoreceptors. The most important photoreceptor is phytochrome, which controls red light responses, and blue light response are controlled by cytochromes and phototropins. These receptors regulate the process such as phototropism, stomatal opening, and circadian rhythms. So basically, plant photoreceptors are phytochrome, cytochrome, cryptochrome, phototropin, and UVR 8. Red light response. It is controlled by phytochrome. Phytochrome exists in two forms. PR is the inactive form and PFR is the active form. When red light converts PR to PFR, far red light converts PFR to PR. So phytochrome functions as seed germination, shade avoidance, flowering induction, and re-etiolation. Blue light response. Blue light receptors include cryptochrome and phototropins. Blue light regulates phototropism, stomatal opening, chloroplast movement, and inhibition of stem elongation. So this class we learned how plants capture light energy through their antenna complex, convert it into chemical energy, fix carbon dioxide, and regulate their growth through light controlled mechanisms. Understanding photophysiology helps us understand how plants adapt to different environmental conditions. If you both found this lecture helpful, like the video, share it with your friends, and subscribe to Biology at Priyajit for more plant physiology classes. Thank you for watching. So keep in touch. See you in the next class.